morning, everybody. I'm very honored that I may give the first presentation today. So I have tried to make an introductory talk. It was not easy. There's a lot ongoing. And actually, there are more slides that I put out than that I put in. So I hope I can make it in about 25 minutes or 20 minutes. So my talk has three main points: getting started with blood EV research, the challenges we encounter, especially the sample complexity, detection of EVs. I will give a few examples of old but also new problems. And I hope to give a sort of look into the crystal ball by telling you what I think may be future developments. Now, some of you may know that this is not the first blood EV workshop. In fact, the first one was 2 years ago, organized in Helsinki by Pia and Sarah. It was a very successful meeting, and I think that's the reason that we are continuing this, and it's starting to become a sort of tradition. So this is in fact the second blood EV workshop. Now, there was one thing, and this is a quote from a paper I come to on the next slide. One of the things we heard in Helsinki was that starting blood EV researchers found it very difficult of getting started, finding relevant and new information. Difficult to start research, avoid pitfalls, etc. So where to start? So this was the reason. It took a while, but beginning of this year, Pia and I published a paper, and Jeff. It's called The Beginner's Guide to Study Extracellular Vesicles in Human Blood Plasma and Serum. And it's especially aimed at people studying blood EVs. And it contains many different sections. I will just show you two. But one is we updated all the guidelines. And for example, this is one of the parts showing how to prepare human blood plasma and serum. But we also have a glossary. One of the things I noticed, or we noticed, also as reviewers of papers, is there's always a lot of miscommunication about terminology when people are talking about anticoagulants or whatever. So we have a sort of glossary here trying to explain all commonly used terms, etc. This paper, the guidelines, is based actually on an older paper, which is much more comprehensive, but it's a paper from 2017. I think most of what we wrote down in 2017, all the recommendations and considerations, I still think they hold true. The paper we published last year, or this year, Beginner's Guide, is just an update. But I think if you want a complete picture, you should look both papers. Now, getting started, it's not only reading. I think you also can reach out for information. ISEF has a rigor and standardization subcommittee, and within the subcommittee, we have multiple task forces. And if you press the button lower left on task forces, you will come at the task force. And one of the task forces is the Blood EV Task Force. The email addresses are there of the members of the Blood EV Task Force. Output is there. And if you want information, you can also always contact me or one of the members. Importantly, I think that was a slight mistake a bit earlier, the new chairs are Fabrice Lucien Mattioni and Dakota Gustafson. So I stepped down and they're the new chairs. Also, when you want getting started, do not forget that ISEF has online 3 massive open online courses. There's a lot of information. This is the last one on detection isolation of intact vesicles. There is a lot of information present on the ISEF website, so use it, please. Now, challenges. I think the biggest challenge we are facing when you would like to work on blood vesicles is the sample complexity. And I would like to explain a paper published about, I think, 2023. It's called My Blood EV, and I will try to explain why I think this is a relevant paper. So when you talk about sample complexity, it's always a bit difficult to envision this, but just suppose that EVs are screws, just to make it simple. Now, if you look at the left, this is what your sample is. You are not only facing screws, not only having screws, but also nails, bolts, nuts, etc. So the screws are only a minor part of the entire mixture. And actually, this is precisely what is with your blood plasma and your blood serum. If you not sure whether you can see my mouse. Yeah. If you look at the top right here, you can see a few vesicles. This is plasma, normal human plasma, scale bar 200 nanometers. But what you see is there are many white particles around. These are all lipoprotein particles. So keep in mind that if you're measuring vesicles, they are present in a soup of non-EV particles. And it's easier to measure the non-EV particles than to measure the EVs. So now the interesting thing is that we know that blood is very complex, but when you look at collection, handling and storage of plasma and serum, we all have different protocols. It's completely not standardized. We all do a different thing. And that's causing problems. And this was beautifully illustrated. I think this is a brilliant paper. It was published also last year, so a very recent paper. And what they did is they would like to explore how different laboratories preparing biological samples like blood and urine, what are their protocols, what are they doing? Now they sent out surveys to JIVEC members, so they're the members of the Spanish EV Society, and they got responses back, 70 surveys, and the variability, and this is a quote from the paper, in pre-analytical approaches reaches 94%. So when you look at collection, handling and storage, we all do very different things. Now, and why is this? So we all have different protocols, different centrifuges, different operators, etc. So why is this? Well, I think, to be honest, I think it's the sample complexity that's causing this. We have too many pre-analytical variables. We have at least 37 pre-analytical variables that can potentially affect the concentration of vesicles, the composition of vesicles, the function of vesicles, etc. So it's really very complex. What can you standardize? Where to start? So at that moment, it's time to look for help. Actually, it resembles me a bit, to be honest, if I look at this. But so we need help, and this is where what I think the cavalry comes in. It's always a bit late, as you know. But the cavalry in this case is, I think, the ISAF Rigor and Standardization Blood EV Task Force. So this is the paper they published last year, My Blood EV, Minimal Information to Enhance the Quality and Reproducibility of Blood Extracellular Vesicle Research. Now, what is the idea of this paper? The idea of this paper is, and I think this is one of the most important messages of my talk, I think if you prepare blood plasma or serum, the sample has a very central key role. And what do I mean by that? Well, on the one hand, you collect blood, and you have a protocol, an operator, and equipment, and you have your end product, and the end product is plasma or serum. This is the end product of the pre-analytics. At the same time, the same tube of plasma or serum is the starting point for your EV research. So you start to isolate and detect vesicles, you look at the biochemical composition of vesicles, and you try to functionally characterize these vesicles. So, as I hope I showed you on the left, we have completely no standardization. Then we all try to detect a very small fraction present in a very complex fluid, and then we all expect reliable, comparable, and reproducible results, which often is not the case. Now, what is My Blood EV? It's very simple. If you write a paper where you do EV research, you always have a method section that you have to write, and you have to write down the protocol of blood collection, preparation, storage of plasma and serum. This is what everybody's doing, or should be doing. What My Blood EV is that I think we should go one step further, and also, if possible, try to measure and report whether you have hemolysis, the lipoprotein concentration and the platelet concentration. For example, the platelet concentration is a measure for the efficacy of your centrifugation procedure and how well the operator has been able to pipette off the plasma without platelets. You can find this checklist, there's a checklist at isaf.org. And so I think the important thing is that with My Blood EV, we do not choose for standardization of these 37 variables. You can use your own protocol. So ISAF does not tell you what to do. I think it's important. You, as a researcher, you have your own freedom. But please measure and report the quality of your samples. Why? Well, I think at least 3 different reasons. I hope that in a couple of years we can generate data together, and I hope that we can use these data to develop evidence-based protocols, for example, collection, handling, and storage of samples. I think also we can independently inclusion and exclusion criteria for samples. They should be included or excluded in a certain downstream EV analysis. And I think we could this could enable the exchange of samples between researchers and biobanks. Now, every biobank is a standalone system, and the quality of a biobank for one biobank is completely not comparable to the sample composition of other biobanks. So what is the advantage, I think, in the long run? It's improving reproducibility. So it's good for the comparability, but also the credibility of blood EV research. So in very short, use your own recipe, use your own equipment, your own oven, but report the taste of what you have produced. I think this is, in a sense, what my blood EV is about. Okay, detection of EVs. I also had slides about isolation of EVs, but I think it was way too much, so this is one of the things I let out. But I would like to give you one example that problem that we are facing recently. It's unpublished. So in 2017, Jens P. Simonson asked the question, what are we looking at? Extracellular vesicles, lipoproteins, or both? And I think it's a good question. It's a very fair question, because as I showed you, lipoproteins greatly outnumber vesicles in human blood. Again, same picture on the left, lipoproteins all over the place. And I think it's also nicely illustrated here. This is a picture from the compendium on flow cytometry published in JEV. You see here the concentration on a linear axis and the diameter of the particles measured. In blue, the area below the curve are the lipoproteins, so the estimated size distribution, particle size distribution curve of lipoproteins. And here in black, these are the vesicles. Now, we do not precisely know the concentration of vesicles, but this was a sort of consensus among the 20 or 30 people writing this paper. But you see the fraction of vesicles is way lower than the fraction of lipoproteins. And especially if you go below 100 nanometer, it's incredible how high the concentration of lipoprotein particles is. Now, this is also, I think, beautifully illustrated by already an old paper from Rebecca Dragovic. I think she was the first to publish data by nanoparticle tracking analysis. And what you see here, this is the concentration of vesicles on the y-axis. Actually, it's wrong, it should be particles. And here you have the diameter of the particles that are being measured. Now, when you measure with an NTA, you measure all particles, and this is the light scattering profiles you got. But these are mostly lipoprotein particles. So if you add here, and they added a membrane dye, it inserts into the membrane of vesicles, and then you see that there's only a very tiny fraction of particles that stain positive for this membrane dye. In other words, vesicles are really a small fraction of all the particles present in plasma. In fact, with the knowledge we have now, we know that I think the lower limit of detection of NTA is about 80 nanometer, meaning that the left part of these curves actually are not existing at all. This is just showing that your lower limit of detection is there. Now, why do I show this? Well, this is data unpublished. We're writing the paper right now, but I think it's a finding that may hopefully set your brain at work. At least it set our brains at work. So these are points. All these are points. Every point is a measurement of data from a sample of the effects study of Alexandra Gazechka. It's a clinical study, completely not relevant. But Edwin plotted the concentration of all particles measured in plasma samples on a logarithmic axis here on the x-axis. And here's the lipoprotein particle concentration. Now, you see there's a massive, very nice correlation between the concentration of lipoprotein particles and the concentration of all particles, which is not surprising. So the more lipoproteins, the higher the concentration of total particles. Now, the interesting observation is this, that if you plot the concentration of all particles, again, just as here on the x-axis, here Edwin plotted the concentration of labeled particles, so with antibodies with a fluorochrome. This is the concentration of labeled particles we measured. Now this is selected here in positive particles, so PS exposing particles, platelet particles, red blood cell vesicles and endothelial vesicles. But what you see is, for example, especially for endothelial vesicles, you see there's a very strong correlation between the total number of particles and the concentration of labeled particles. So the concentration of endothelial vesicles is very low, but there seems to be a very steep relationship, a very nice correlation with the total number of particles, suggesting that perhaps we are staining lipoproteins here rather than vesicles. And this also holds true to a certain extent for red blood cell and platelet vesicles. And in fact, if we correct for this, we see in all samples more or less the same. That's an interesting observation. So what do we measure, lipoproteins or vesicles? We all think that adding an antibody against a CD marker is a good way to distinguish lipoprotein particles from vesicles. I think based on this, perhaps we should reconsider. Future developments. Well, I have to admit, part of this is just what I think, but it's my opinion. So if you don't agree, it's fine. So I think the most important question I still would like to answer is, what particles are really out there? So if you have a blood sample, what particles are really present in the blood? Now, this is a particle size distribution of vesicles in a conditioned culture medium. It's not so important. Data from Augustine. Here you see the diameter of the particles measured, and here you see the concentration on the x-axis. Now, I think when I look at what we and all other people have been publishing on vesicles, I think I would like to put in two question marks. I think, first of all, we have not really an idea about the smaller vesicles. We have some very sensitive detection techniques. They are low throughput. We don't really know what's out there. On the other hand, and I put this in especially for Dolores, I think we also, when I look at plasma serum, I don't know what is out here, because well, if you put here a logarithmic axis, you will see we do have a few large particles, but the concentration is very low compared to the small particles. So what is really out there? I will show a few slides first on the small ones and then on the big ones. Now, if I ask the question what small particles are out there, I think a number of studies have been published during the last 3-4 years, very interesting, where they looked with cryo-electron microscopy in different fluids, also body fluids, what types of particles are there. And one of the papers, and I think they show beautiful pictures, this is from human cerebrospinal fluid. You see all sorts of different particles in CSF. You see filled particles with a single membrane, but you see particles with a double membrane, even particles with 3 membranes. Are these all vesicles or not? Particles in particles, etc., etc. So you can also ask the question, what small particles are present in normal human plasma? So if I collect blood from normal healthy humans, what small particles are out there? So what Augustine is doing in Leiden, and I really like his work, he is trying to make a comprehensive small particle atlas, EV atlas of human plasma. So he has developed a high throughput system where you can analyze up to 50,000 particles automatically, and you have AI-assisted detection and classification of the particles that you measure. So you can see whether the particles are lipid bilayer, the morphology, size, contents, etc. This is right now ongoing. I can show you data. It was developed for conditioned culture medium, but now we're doing this for plasma, and we're trying to generate really a sort of atlas of trying to get a feeling what is out there. We see many particles we have never seen before. So this is work in progress, but I think it's interesting that we get more insight into the small particles. Now, what big particles are out there? And I must say, I don't know anything about big particles, but there has been an ISAF workshop. I haven't been there, so perhaps what I tell you has been shown there, perhaps not. But I was thinking about this from the blood perspective again. Now, the protocol and I think it's a mostly used protocol used by people in the field that start working with blood. You centrifuge blood 2 times to remove the bulk of the cells, etc. Now, this protocol was published by Lacroix in 2013. It's a paper, I think it's really well done work. But 2013, if you see at the progress we have made during the last decade, it's a very old paper. Now, when you look back at what they have done to look at the presence of vesicles, to be honest, I think they never detected vesicles at all. It's with the knowledge we have now, but we do know from a 2018 study that the sensitivity of their flow cytometer was insufficient to detect vesicles at all. They could detect platelets, that's about it. The gating strategy using polystyrene beads of 500, 900 nanometer, also that confirms they were measuring platelets. So we also do know, and I show you 2 examples, next slides, not all cells and not all cell goes are removed by this protocol. And even contamination with a few cells can have a strong impact on your downstream results. So, and also, and I think this is something I never really thought about before, but this protocol was removed to remove cells, but it was not to develop to preserve large EVs. Now, if you use this protocol, and we do this really to the best of our abilities, we still have cells there. So after 2 sequential centrifugation steps, 1 centimeter Buffy coat repipette, etc., we still have activated platelets. These are data from Jill Bracht and Mendel Engelaar. But we also see red blood cell goes there. And keep in mind, you cannot easily separate these from your vesicles when you do a downstream analysis. So when you are looking at the vesicles, these will also be present, and they are much larger than the vesicles you're looking at. So they're elephants in your population of mice. It's a big confounder, even though you have a few. But perhaps most importantly, what I said is the protocol has never been developed to preserve large vesicles. And I think this is illustrated by a paper we published several years ago. It's work from Linda and Frank Kouwmans. And they developed a model, a physical model, based on the Stokes equation, where you can put in the g-forces, the time, etc. And then you can look at the ability to remove small particles like beads or vesicles or whatever. So the model was validated by reconstituting beads of various sizes into plasma or buffer. Now you cannot read the curves, I think the data are too small to show, but just one of the examples. If you centrifuge for 22 minutes 2700 G, which is about equal to what we do in the first step of the Lacroix protocol, we have about 90% recovery of the 100 nanometer polystyrene beads, which is very good because we would like to recover the vesicles, and we all assume that vesicles are small. At the same time, we remove 50% of the 1000 nanometer beads. So perhaps, and this is something I don't know, perhaps the absence of large vesicles is explained by the fact that we're throwing the large vesicles away by the centrifugation protocol, meaning that the moment we remove the cells, we also remove the large vesicles. I don't know, but I think this is food for thought, something to think about. So, looking into the future, I was thinking perhaps if this is also a successful meeting, perhaps in 2 years' time we should consider again a blood EV workshop, but I'm not sure. Perhaps US, they have some nice venues there. I hope they're big enough. And yeah, thanks. I would like to thank, of course, the organizer, but especially the members of the Blood EV Task Force. I think we've had lots of discussions. It's all not so easy finding your way in this incredible, difficult, tedious business, but I think we have done a good job altogether. I would like to thank the members of the Amsterdam Vesicle Center, in particular the postdocs, Britta, Jill, Augustine, and Edwin. I showed some of their unpublished data as well. And of course, international, national collaborators, especially thanks to Pia for help with the beginner's guide. I think it is, again, it's an important work. So what are my take-home messages? I think please use all the knowledge and resources that are out there. I can tell you from experience, we have done about everything wrong for at least 20 years. and learn from our mistakes and do not reinvent the wheel. I think that would be really, really stupid. I think also as a community and also as a researcher, please support reproducibility. For example, my blood EV and of course also MySEF, etc. I think it's good for your research. I think it's also a sensible use of resources. I mean, you're not wasting money. And being Dutch, I think it's also something we always ask, what does it cost? So don't waste money. It's good for the credibility of EV research, and I think also for ISEF. And I hope I sort of convinced you that I think it's a continuous work in progress. It's very difficult to measure vesicles. They're a very tiny heterogeneous fraction of small particles in a very complex fluid. And at least I'm still learning every week or every month new things. So yeah, keep having an open mind. The field is not settled yet. I mean, it's more difficult than measuring leukocytes. It's really, really a challenge. That's it. Thank you.
Thank you for this very interesting talk, and thanks for focusing on the large EVs, but I'm not asking you a question about that.
Sure.
I wonder, so you said that we have to report hemolysis and platelet counts, and I wonder, like, how do we do that? Because we need the SOPs also for that, obviously.
Yeah, it's so, of course, I work in the hospital setting, and then it's relatively easy because we have all the equipment available. In the My Blood EV paper, we give a few options how people can do it. I think it's important to realize that in some studies, like existing biobanks, you will not always be able to do this because the samples are already there, or you have very valuable clinical samples. I think that by itself is not a problem, but at least report you have not done it. Why? Because I hope that we can see in the long run, I mean, not in a year, but perhaps 5 years, that if people are studying, for example, microRNAs that are associated with plasma EVs, that we see that the people removing platelets or having low platelet counts in plasma have more reliable data than the people who have high platelet counts or do not report anything. And I think that's sort of the underlying goal. Again, if you cannot do it, it's fine. It should not hinder you from publication. But I think as an easy quality parameter, it would really help the field forward. And I think it's good to mention that it's a simple idea from a clinical perspective, and it's picked up also by for example, the ISTH, which is a very clinical society. We submitted a sort of supporting paper there, which they would like to upgrade to formal recommendations because they think it's a way forward, not only for the EV field, but also in general for the biomarker field or the biobanks. Because if you implement these from a clinically perspective, simple lipoprotein concentration measurement or platelet concentration measurement, your sample becomes much more valuable than without this additional information. I mean, it's really a sort of quality tag that you can put to your sample. And then, for example, you can say in the end that if your platelet count is larger than, let's say, 5,000, you will not use it for a microRNA analysis because it's not suitable. But we do not have these data right now, so we need a sort of bridging period to generate a database with information there. But the fact that also helps the European Liquid Biopsy Society is very much likes this idea because you get better biobanks. And I think, again, in the long run, it may help to really make progression with the field rather than concentrating on standardizing these infinite number of variables, because we're going to lose that war, I'm afraid.
Thank you.
Right. Young Park from South Korea KAIST. I have a question about your flow cytometry-based detection of the EVs. You showed a very good positive relationship between the total particle concentration and the lipoparticle concentration, but that was not the case for the EVs. And you mentioned that the possible reason for this result could be that the staining we are doing for EV might also stain the lipoproteins. But remember that the PowerPoint slide you showed us stained CD63 or CD61 positive EVs. So I was wondering whether you used immunostaining for detecting those kind of EVs or other staining method for detecting the EVs.
No, that's a good question. So these were just observations that we are just trying to figure out what sort of artifacts are there in our measurements. And I think now we have seen this, and this is only part of the data that I showed because it's work in progress. We didn't publish this yet. But at least it made us think that perhaps we are staining lipoproteins, we have false positive lipoproteins, and now we are setting up a new series of experiments trying to validate that we're really staining lipoproteins. I mean, this evidence by itself is insufficient, but it's very, suggestive that there may be again another artifact present in the data that we measure. And again, keep in mind, we've been doing this for years, always being unaware of this, but there's a striking correlation, especially for what Marco would call the rare events for endothelial vesicles. It really seems that being positive does not mean that you're a vesicle.